So here we are again. It's the last of our four major classes of biological molecules, nucleic acids. And in the previous lecture, I tried to build the whole thing, the whole story around protein shape and how the shape is really important to the function. It's not a big cluster of nutrient monomers. This is actually something that has got a whole lot of information that's required in order to make this legitimately very sophisticated thing. A protein has to start out with a very specific sequence of amino acids, okay? Now, I'll oversimplify things a little bit, but for right now, I want you to think of nucleic acids as being all about just the information, right? In the case of the protein, the sequence of amino acids was kind of like the starting point. That's how you ended up. Remember, we call it the primary conformation. And then from there, we got the secondary and tertiary and sometimes the quaternary conformations. And there was a lot more. What I'm trying to say is there's a lot more than just the sequence of amino acids. Okay. Now, in the case of DNA and RNA, I'm going to say it's just about the sequence. The sequence is the whole deal, right? And the reason for that, as we'll be learning a lot later on, but for now it suffices us to know that in the central dogma of information flow in biology, things start out encoded in the form of DNA, and then that code, that information is passed on to RNA, and the RNA uses that information in order to make the polypeptide, the protein sequences. The amino acid sequences in the proteins are encoded in a molecule of RNA, messenger RNA, and that messenger RNA is copied off the DNA. And so while the protein eventually has to take a shape, the DNA and the RNA, it's really just about the sequence, right? And so that's one major point for you to take hold of. The second major point is that we don't have 20 amino acids to work with. We only have 4 letters, right? In DNA, the letters are A, G, T, and C, which code for which are symbols for the bases. There are nitrogenous bases: adenine, guanine, thymine, and cytosine. Those are, that's where the A, G, T, and C come from. And when you have a whole long strand of polynucleotide, it's the sequence of bases that is going to be the information. It's going to carry the information that will ultimately be required in order to make the sequence of amino acids that are there in the polypeptide, right? So again, getting back to the central dogma, the informational flow, we need to think about how a protein's amino acid sequence is being encoded, and it's basically encoded in the form of RNA and DNA. The base sequence of DNA and RNA is what specifies the amino acid sequence in the protein. You'll hear this many, many, many more times, right? And then we're going to be reviewing the structure of a mononucleotide very shortly. In fact, we can go ahead and do that now. If we go over and look at the chapter 3.15, which is one of your readings for today's lecture, it gives you the basic structure of a nucleotide, right? Now, this is a mononucleotide. Recall back from previous discussions, I actually drew something like this for you before. Okay? And right there at the core of this molecule is a sugar. For DNA, it's deoxyribose. You might note that there are 6 carbons in glucose, but there are only 5 carbons. Carbon 1, it's a carbon there. Carbon 2, C2, C3, C4, and the 5th carbon is one right here. So we would call this a pentose Sugars, I mentioned this before to you in a previous lecture, and the two kinds of pentose sugars that we care about. One is deoxyribose. That's the sugar you're looking at here. And the other sugar is just ribose. The difference is whether or not this structure there, that hydrogen is just a hydrogen for deoxyribose, whereas for ribose, it's an oxygen. There's a hydroxyl group there. So yeah, DNA is just a little bit more nonpolar compared to RNA because it has one less hydroxyl group. Okay. So sugar is part of it. Phosphate is another part. Sometimes we draw it as a P with a circle around it. That's how we did it before. And the nitrogenous base, they're coming off the number 1 carbon. The one that is shown here is the adenine, so this would be A. Alternatively, you could have G, C, or T in the case of DNA nucleotides. Right? So the bases are a little bit different for ribose. The sugar is a little bit different for ribose, but right now we're only going to be focusing on DNA. Okay. Now, when these molecules come together, we get what the book identifies correctly as a sugar phosphate backbone. Okay. So read here where it says sugar phosphate backbone, and ultimately it comes out kind of simplified in a simplified drawing. You might show the backbone. That's this thing here. This is the backbone. Backbone. Well, the bases are coming off to the side. Okay, and so what I just want to do with you now is to make a connection between what we were looking at before, the sugar phosphate base. Right. So we've got the base over here, sugar, and the phosphate group. This would be one mononucleotide. And when these things get strung together, you end up with mononucleotides attaching from the sugar to the phosphate of the next mononucleotide. It's the base. And that's going to hook up to the next mononucleotide, the phosphate. In other words, what we're looking at in any of these depictions is really the same thing. And so this base might be cytosine, this base might be thymine, this base might be adenine. And so you've got the mononucleotides. This would be a mononucleotide right there, right? Because you see the sugar, the phosphate, and the base, and they hook up one to another. This is how we end up with a polynucleotide, right? Et cetera, dot, 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 right? And so this part here, phosphate, sugar, phosphate, sugar, phosphate, sugar, that's the backbone, sugar, phosphate. And the nucleotides, the nucleotides bases are the things that are hanging off the side. This is just another way of depicting. In the textbook, what I feel is like sometimes there are so many different ways of depicting a molecular structure. And in the case of DNA, we've got such huge molecules to deal with. It's important for us to do some shorthand. Otherwise, we get bogged down by all the atoms and bonds that are internal. And so it's a good idea for us to do that. But sometimes it's hard for students to have retain the vision that we're always talking about the same molecule. Okay. Now, the other thing about DNA is that it is a double-stranded molecule, right? So the characteristic feature of DNA, it's double-stranded, and also forms a helix, right? A helix is a coil. And so the way that you can sort of visualize this is we've got two strands. One strand is, this might be strand number 1. So strand number 1 is forming a helix. Strand number 2, do it in a different color, so it'd be in red, is forming a helix. And they kind of wrap together in this double coil. Okay, so sometimes it'll be drawn like this. So I usually draw it like this, not quite as artistic as and accurate as what we have over on the left. But the general idea is that there are 2 strands always, and one of the strands is, one of the strands will carry the information. The other strand will just be occupying the space opposite of the DNA strand, of the first DNA strand. Okay, so maybe this is a blue strand, and if we unwind them, if you actually unwound this, if you unwound this, and pretend like we don't have wound together DNA strands, but you separate them out, what you find is that on one strand we've got a T, an A, a G, and on the other strand, on the red strand, you would have an A, a T, and a C. Right, and so another feature of this. Okay, so let me put all these buzzwords on the board for you so you can see them. 1, double-stranded. We're talking about DNA here because RNA, as we'll find out shortly, is not double-stranded. It forms a double helix. Which is a double-stranded molecule. It kind of looks like a ladder, and it is spiraled. It's coiled up on itself. And each strand is that structure of a sugar phosphate backbone with the bases coming off to the side. But what's going on with the DNA molecule is we've got this phenomenon called complementary base pairing. Complementary base pairing, in which we always have 2 bases that complement each other. If one strand has A, then you must have a T opposite, and vice versa. If you have a T on one strand, you have to have an A opposite of it, right? If you have a G on one strand, you have to have a C opposite of it, and if you have a C on one strand, opposite of it, you'll always be finding a G. These are the rules of complementary base pairing. If you've learned these in a previous biology class, it's great. If you haven't learned that A always pairs with T, T with A, G always pairs with C, C with G, now would be a good time to just memorize that, right? These are the rules of complementary base pairing. This is how DNA molecules are held together, right? And so the next bit is, well, why do we have complementary base pairing? And how does it hold the 2 molecules, the 2 strands of DNA together? And it's a good thing you asked me that because we've already answered that. As it turns out, the A and T have delta plus and delta minus charges on them, okay? In the structure of adenine, we've got delta plus and delta minus charges. And on the structure of thymine, we've got delta plus and delta minus charges. Only things are set up so that if you have the 2 bases, A and T, on opposite strands, these 2 structures fit together not only structurally, but they also have these delta plus and delta minus charges in exactly the right location so that we can get an attraction between the delta plus and delta minus. What are we talking about there? Okay. What is the name of that attraction between delta plus and delta minus charges? Okay. Yeah, I hope you said it's a hydrogen bond. Okay. If you want it to be more accurate, you could call it a dipole-dipole interaction, or a weak interaction, a weak ionic interaction. Those would all be somewhat accurate. And so that's the case, this is the reason why we have complementary base pairing between A and T, right? If you look at a G and a C, yeah, they also fit together, and we also have delta pluses and delta minuses. Only in this case, we've got 3 points in which the delta pluses and delta minuses are exactly oriented opposite of each other. And so there are 3 hydrogen bonds that hold together the G and the C. So completely accurately, you might have hydrogen bond there, another one there, another one there. Whereas with the A and the T, you'd only have 2. A and T only has 2 hydrogen bonds. G and C has 3. Right? But the key ideas here is that, well, one, complementary base pairing is a thing, and 2, the mechanism underlying the complementary base pairing is hydrogen bonds, and it's also the force that keeps the 2 strands hybridized and tightly right up one against the other, right? So when we make a molecule of DNA, we're actually making 2 strands that pretty much carry the same information. We just have a complementary strand opposite of the other one. So if these are hydrogen bonds, they are what keeps the DNA molecules zipped up nice and tight. Okay, so let me go back to my outline, see how we're doing as far as my covering of everything we need to cover, right? So now it's all about the sequence, all about the information. We will be talking about other functions of polynucleotides later on. 4 letters of DNA create a base sequence. Sugar, phosphate, and a base. Mononucleotides become polynucleotides through bonds between sugar and phosphate, which give us a sugar-phosphate backbone, and the bases A, G, T, and C are hanging off of the side. DNA is double-stranded, and the 2 strands always have complementary base pairing. A always pairs with T, T with A, G always pairs with C, and C with G. Okay? And the bases match up. Okay? When you've got them on opposite strands, they match up delta plus and delta minus starches, giving rise to hydrogen bonding, and that's the force that keeps the 2 DNA strands together. Okay, now on to RNA. RNA is pretty much the same deal, only in place of deoxyribose, right? We have ribose, right? Deoxyribose, it's a different sugar. In other words, we have that OH on the number 2 carbon, which is something I don't really care that you know. We've got a phosphate group, and we've got the bases. Okay, the base hanging off to the side, maybe A, it might be G, it might be U, which replaces the T. Oops, that should be U. Yeah, there's no, there is a cytosine, but I didn't mean to write a T. And the last one is going to be a C. Okay, so no uracil, you have, I'm really messing up here. So let me try that one more time. Right. So in RNA, you don't have thymine, you've got uracil. And so A, you've got G, you've got U, and you've got C. These are the 4 bases that go into RNA. Okay. Another thing about RNA is that it tends to be single-stranded. It's a difference from DNA, and functionally, RNA is going to be produced. It's going to be carrying the information that's there in the DNA, and tends to be a much less permanent, less shorter-lived structure. Right now, the reason why RNA is of some significance to us in our post-pandemic world or mid-pandemic world, depending on what view you take on this, is that the Pfizer and the Moderna vaccines are mRNA viruses, or sorry, mRNA vaccines. We're entering a new era in which the RNA is going to be used as the actual challenge. We're challenging the immune system with RNA as opposed to what we've always been doing, which is to challenge the immune systems with the actual proteins or with the actual antigenic targets that immunity has to be built up against. Okay, but that's for a future discussion, but definitely RNA is within our consciousness in this modern world. Okay. Apart from that, it's pretty much the same. The last thing I think I wanted to talk about was to return to an idea that I gave you before, which is that if you look at the structure of when we go to make polynucleotides, DNA and RNA synthesis, anabolism. Okay. When we make DNA and RNA, we don't start off with simple mononucleotides. We start off with mononucleoside triphosphates. Okay, I'm just going to say mononucleotide triphosphate. Technically, from a chemist's point of view, the appropriate word there would be nucleoside, and it's not that big of a deal. Let's keep things consistent. But the reason why we're starting off with triphosphate, anybody remember that? Yeah, that's exactly right. Student, imaginary student, whom I'm sort of visualizing answering the question, which is that when you have a mononucleotide triphosphate like A, when you got that adenine here, I showed you in the picture from the textbook, and the phosphate, this would be the phosphate that's part of the mononucleotide on its own. Okay, when you add a couple of more phosphate groups, okay, this is going to be pre-charging. I called it a pre-charged monomer unit, and that allows us to undergo the process of DNA and RNA synthesis without bringing in an outside energy source. Okay. Now, there's a reason why I belabored that point earlier, and it has to deal with another key concept that we'll be getting at very shortly. If this is actually ribose, and this is actually adenine, and you got 3 phosphate groups on it, that would make this a very special molecule. It would be adenosine triphosphate. There's an O there, but get the idea. Or ATP. Right? ATP is going to be a very significant molecule for us. This is the currency that is used for all cellular reactions that require energy input. Remember, I gave you the idea that some reactions are exergonic, and some reactions are endergonic. Okay. And exergonic reactions do not require an outside energy source because energy is released. Endergonic reactions, however, are not going to be moving forward. An example of an endergonic reaction would be dehydration synthesis, glucose and glucose coming together to form maltose, and you end up with a water molecule, right? Remember dehydration synthesis? You should. It's going to be on the test, right? So an endergonic reaction requires an energy input. Where does the energy come from? Well, the energy is almost always going to be coming from an ATP molecule, right? ATP is the currency of energy within the cells. All cellular reactions that require an energy source are going to be using ATP as the energy currency. We can leave that point right now, and we'll come back to it after the break. After the exam, we'll be in unit 6 or 7. I don't remember which one it is. But now we're going to go back to our textbook and see where it takes us. Okay, a function of nucleic acids, genes. We talked about this. Here's our central dogma concept: is that the information to make a protein is stored in DNA, and RNA is the molecule that's used to carry the information from where it is stored in the nucleus, that's DNA, to where it's actually needed out in a different compartment of the cell and it gets converted, it's translated into protein. Okay. There's a nice little video of a hipster guy that is explaining DNA structure to you. And there's also a chapter 3.16. I'm not exactly sure how this is relevant. I mean, it's a nice story. It's a great story. Yeah. Why do humans, among all mammals, have this weird characteristic of retaining the ability to digest lactose into adulthood? And in fact, not everybody can do that, right? There's a lot of people that are lactose intolerant. And lactose intolerance, the inability to digest lactose as an adult, is really completely normal, right? You're supposed to lose your lactose tolerance after you stop drinking milk, which is sometime in early childhood. Okay. Now, it's just for these weirdo human cultures that started depending on milk, on cow's milk, yak's milk, camel milk, as a source of sugar, as a source of drinkable liquid, that required people to deal better with lactose as adults. And so that story is developed here, and it's a mutation away from the normal state of lactose intolerance to lactose tolerance. And oh, that's a great story. I like the story, but I honestly don't see how it's completely relevant to our discussion of the structure of DNA. So I'm going to go back to my whiteboard and look at our board here. We completed the discussion of RNA. The 4 letters of RNA are A, G, U, and C. I had a little bit of difficulty with that U, but you got it, right? You don't have to recut the entire video just to correct that little thing. Okay, the basic structure of RNA is the same, sugar phosphate backbone with side bases, although we don't have the double-strandedness in RNA. Okay. Central dogma, we've been through that a couple of times. And the last bit was to connect our discussion right now with the structure of nucleic acids to a discussion that we'll be having very shortly when it comes to understanding the roles of ATP in cellular dynamics. And that is it for today's lecture. I hope it was clear enough. See you guys soon.